Recall that bacteria have a semi-permeable phospholipid bilayer that makes up either the cytoplasmic or cell membrane and also the outer membrane in the case of gram-negatives. There are two features to these membranes. The first is the phospholipid bilayer, and the second are proteins, either peripheral or integral proteins, that are actually bound or somehow anchored into this phospholipid bilayer. In the case of the phospholipid bilayer itself, I want you to recall that there are a couple of features of these. We have two fatty acid tails, a glycerol molecule, and then a phosphate group which can have modifications on it as well. And that's just shown up here as well. So we have, again, those two fatty acids, 1 and 2. We have the glycerol molecule, which is a 3-carbon carbohydrate. The phosphate head group here, and again, sometimes this can be modified with additional attachments onto that phosphate. The structure of this membrane then is such that we have those phosphate head groups interacting with the aqueous solution that the bacteria would be found in, and also interacting with the cytoplasm, which is an aqueous solution as well. Those phospholipid tails then point toward the middle or in between these phosphate head groups, creating that selectively permeable barrier. Now, bacteria are unlike humans in that their internal temperature is in part going to be based on the environment that they're found. And so what that means is that there's going to be changes to the phospholipid bilayer to maintain homeostasis and to maintain fluidity as temperatures fluctuate. So for example, as temperatures increase, that's going to increase the movement of the phospholipids. And in order to reduce then the fluidity and to reduce the movement of molecules through that plasma membrane, bacteria will increase the amount of saturated fatty acids within that membrane. As temperatures cool, that's going to slow down movement. And that slowing of movement then means that there needs to be more space between those phospholipids in order to maintain fluidity. And so what we find, one way to create space is to increase the concentration of cis unsaturated fatty acid tails. And you may recall from our conversations before that these cis fatty acid tails create kinks, and that creates spacing between the phospholipids. On the other extreme, there are some archaea that live at very high temperatures. Archaea that we would classify as hyperthermophiles. And in order to maintain, again, homeostasis and a consistent membrane fluidity, what we find with some archaea is that they actually have a monolayer, where we have modified fatty acids that are not only branched, but you see here are associated with glycerol molecules at each end. And so in creating a phospholipid bilayer, this creates a phospholipid monolayer. Though some molecules can make it through the phospholipid bilayer, many of them cannot. And so we need methods of transport. And so in the next few slides, we're going to talk about modes of transport of molecules across the phospholipid bilayer, focusing on the three listed here, particularly simple diffusion or passive diffusion, the two terms are synonymous, facilitated transport, or active transport. Now, some molecules can move through the phospholipid bilayer by simple diffusion, and molecules that can do so are typically small and nonpolar, such as the gases oxygen and carbon dioxide. Because these are nonpolar molecules, and because of the fact that they are small, they can easily diffuse through the phospholipid bilayer without any sort of transport mechanism. In this method of movement through the phospholipid bilayer, these molecules are moving from areas of high concentration to low concentration. In facilitated transport, this often occurs with molecules that are polar or a bit larger that cannot pass through the phospholipid bilayer easily. Water is a great example of a molecule that requires facilitated transport. The transport of water through the phospholipid bilayer requires molecules or proteins that are known as aquaporins. And as the name would suggest, these are pores of proteins that create a channel that allow for water to diffuse in or out of the cell. And this movement of water requires no energy whatsoever. And that's what makes facilitated transport different from active transport, which we'll talk about in a few slides. Now, the movement of water through these aquaporins, or these protein channels, is going to be dependent on osmotic pressure, and we'll talk about that in the next slide. Water moves through aquaporins from areas of low solute concentration to high solute concentration. And the movement of water is across the plasma membrane is trying to reach equilibrium so that the concentration of the solute is the same on both sides of that plasma membrane. Now we can define the relative concentration of solute between two solutions, and the terms that we use to define these are isotonic, hypertonic, and hypotonic. And again, these are comparative terms. An isotonic solution is one in which the solution outside of the cell, as shown in the left over here, is the same as the concentration inside of the cell. In both of these we find that there is 20% solute concentration, 80% water concentration, and so the movement or net movement of water in and out of the cell would be 0. In the middle, we see an example of a hypertonic solution, one in which the solute concentration inside of the cell is 20%, water concentration is 80%, and the solute concentration outside of the cell is 40%, and therefore the solute concentration outside of the cell is greater than the solute concentration inside. Water is going to move out from these cells in order to reduce that percent solute concentration, again to reach equilibrium across the plasma membrane. And so we would say in this case that the solution is hypertonic relative to the cell. In the last example here, we can see an example of a hypotonic solution, one in which the solute concentration outside of the cell is 10%, the concentration inside of the cell is 20%, and so in order again to reach equilibrium across the plasma membrane, water will move through aquaporins into the cell, thus increasing the size of this cell. We'll talk about the implications of this with relationship to red blood cells and to bacterial cells during our lecture. Facilitated transport occurs in the absence of any energy. What we find in facilitated transport is the movement of a molecule with the concentration gradient. For example, let's say that we have a solute here, and you see that the concentration of the solute on the outside of the cell is greater than the concentration on the inside of the cell. In other words, on the outside of the cell there are 1, 2, 3, 4 molecules of the solute. On the inside there is a single one. And so the movement of this molecule is going to be from greater concentration to lower concentration. In other words, its natural movement would be towards the inside of the cell. However, if this solute or molecule is large or charged, it can't get through the phospholipid bilayer, and so it has to pass through a transporter protein. In facilitated transport, what we find are that there are specific pore proteins that will bind to the solute through more electrostatic interactions rather than covalent or ionic binding, and then the molecule will move toward the inside of the cell. We can find these transport proteins being used in one of two methods. Some of them just remain as open channels. Others will actually change shape as the molecule or as that solute is moving from the outside to the inside of the cell. Active transport requires some sort of energy in order to move molecules across the plasma membrane. This image here is just giving an example of 3 different types of active transport. The one that you are probably most familiar with are ones that involve ATP as a form of energy. In bacterial cells and in eukaryotes, we describe these types of transporters as being ABC transporters. The ABC stands for ATP binding cassette. There are ABC transporters that will move molecules into the cell or out of the cell. This is an example of one in which a molecule is bound by a protein, is brought to the transporter, and using ATP, that molecule is moved across the plasma membrane into the cell. An example of this would be the siderophore, which carries iron, which is this little molecule right here. The concentration of iron in the solutions that bacteria are found in are actually lower than the concentrations inside of the cell. And so in order to move those iron molecules against their concentration gradient, this transport requires some form of energy in the form of ATP. Now there are also ABC transporters that are exporters, ones in which the molecules, instead of being moved in, are being moved out of the cell. We find bacteria use this as a mechanism to efflux or to remove antibiotics from cells, and we'll talk about that later on in the semester. There are two other mechanisms of active transport, ones in which we use hydrogen power or a hydrogen gradient and couple the movement of hydrogen with or in the direction of its concentration gradient with a molecule that is moving against its concentration gradient. And finally, we have group translocation, which again, we use some sort of energy, in this case the form of a phosphate, to move a molecule into the cell and to essentially trap it inside of the cell. Now some of you may know that in the process of glycolysis, the first step of glycolysis is to take glucose and add a phosphate to it. Well, that step right there is part of this process, in which glucose is brought into the cell, a phosphate is added onto it, and already we have the first step of glycolysis taken care of. There are other ways to achieve this first step of glycolysis, but this act of transport of bringing glucose into the cell through group translocation is one of those mechanisms.